Our technology was developed for clinical use, and therefore our focus has always been on providing a safe, non-viral approach to loading molecules into challenging cell types with high efficiency while maintaining high cell viability. MaxCyte cell loading technology is based on electroporation. We suspend the cells in physiologically balanced conductive buffer with relevant loading agents and then apply a series of electrical pulses that create a temporary perturbation to the cell membrane, allowing molecules of almost any size to enter the cell very rapidly. And the beauty with the MaxCyte system is that we use one buffer that works for all cell types. In addition to plasmid DNA, we can efficiently load mRNA, siRNA, and even proteins into the many cell types. One of our technology's most unique feature is scalability. We can transfect small number of cells from 500,000 to few hundred million cells by static electroporation, which takes only a few seconds. Larger number of cells between 500 million to about 200 billion cells can be transfected by flowing the cells through the electroporation chamber. Because we apply the same series of electrical pulses for every transfection given the same cell type, we achieve very consistent results regardless of scale. These are the three instruments that we have right now at MaxCyte. The top one is called the STX that can transfect from 500 million to 20 billion cells in less than 30 minutes. And typically this is used for research and development. The instrument in the middle is called the VLX that can transfect up to 200 billion cells. And typically this instrument is used for vaccine production, AAV production, and lenti production, and even antibody production for large scale. So that would be about 100-liter bioreactor at about 2 E6 cells per mil on day 0. The instrument on the bottom is called the GT instrument. This is the instrument that is solely dedicated for gene and cell therapy with all the FDA master file available for ETA filing. Earlier in the presentation, we saw the impressive transfection results that we can achieve with challenging cell types such as iPSCs using the MaxCyte cell engineering technology. This slide illustrates the transfection efficiencies that can be achieved with a wide range of other cell types, including multiple primary cells and cell lines. Note that MaxCyte technology can also be used with insect cells, enabling the direct transfer of plasmid DNA into the cell without the need for baculovirus. Here are some additional examples of stem cells that customers have transfected using the MaxCyte platform. MaxCyte's non-viral approach to cell engineering enables precise control over transgene expression, enabling modulation of complex biological pathway and cellular processes. MaxCyte technology has been adopted by many biopharmaceutical companies because of its versatility. You can use the technology to modify cell genome using technologies like CRISPR-Cas9. Some companies use the technology for making viral vectors and gene therapy like lenti and AAV. Currently, we have 1 commercial immunotherapy, 15+ licenses for clinical development, and 40+ partner programs in the field of gene and cell therapy. We also have many clients that uses the technology to express recombinant proteins like antibodies and antigens with varying scales from about 10 milligrams to multigram amounts. Our technology is agnostic, so you can use any CHO strain of your choice for stable or transient expression. And you can use any media feed combination that you may prefer in your lab. Some companies in the drug discovery field uses the MaxCyte for ion channel and GPCR expression, which are typically challenging to express. We have several other webinars and publications at maxcyte.com showing these capabilities. MaxCyte technology is an excellent addition of tool in your toolbox, and we can provide solutions to difficult to transfect cell lines and difficult to express proteins. In conclusion, I'd like to highlight the key benefit of the MaxCyte scalable cell delivery platform. MaxCyte is a versatile technology that allows you to transfect any cell, any molecule at any scale, from cell genome editing like CRISPR-based to protein expression, and this protein could be membrane or secreted proteins like antibodies. Because the technology is electroporation-based using a chemical-defined buffer, It allows consistency between transfection from a scale of 500,000 to 200 billion cells in less than 30 minutes. The technology can deliver molecules inside the cell very efficiently while maintaining high cell viability. Therefore, you can expect higher productivity for protein expression and decreased development timelines. Finally, we have a team of dedicated scientists from cell and gene therapy to protein expression, and we work with our clients very closely in order to achieve their transfection goals. Thank you so much for joining us in our webinar.